First of all, I would like to thank the organizers of this conference, especially Mr. Parker, for giving me opportunity to present our work at this distinguished conference. And my name, as told, I'm Alexey Fomenkov. I'm PhD. I'm not dentist or like any dental professional, but somehow my life intervened with the dentistry, because during my tenure at Johns Hopkins, I have a grant from the National Institute of Health, and more specifically from the Institute of Craniofacial Disorder and Dentistry. And today I'm going to present a different story. It's called Restriction-Modification Mediated Barrier to Exogenous DNA Uptake and Incorporation Employed by Prevotella Intermedia. Could you give the next slide? Okay, so here's what I'm going to talk about. Why we study restriction modification systems in Prevotella intermediate, what are the restriction modification systems in prokaryote organism, some classification, the next generation smart sequencing is the way to study bacterial metallome, genome and metallome of Prevotella intermedia, 2 strains. It will cover with the 3 slides. And another topic, it's what we find out in this bacteria, it's a cluster regulatory interspaced short palindromic repeats, or known as a CRISPR-Cas system of Prevotella. And go to the conclusion. So, why we study? So the researchers from the Harvard School of Dentistry come to us and ask to help because Prevotella intermedia is a major oral pathogen associated with endodontic infection and also frequently species found in periodontal infection and also member of orange complex supporting destructive inflammatory disease. In addition, Prevotella also increase impacts on extraoral disorders like nasopharyngeal and interabdominal infection, as well as affect cystic fibrosis in lung. And nevertheless, we don't know anything basically about the virulence mechanism of this bacteria in human infection because there is no genetic system for this bacteria. And it was very difficult to transform or apply any modern molecular biology technique for this bacteria. So to develop functional genetic tools such as the plasmid, transposon, or permit genetic manipulation of Prevotella, we need to overcome this genetic barrier. And basically, I will talk about the results which was published in 2017 in a PLOS One. It's our paper. Next slide, please. So what is the restriction modification system? In 1978, 3 researchers, Arbour, Nathans, and Smith, received the Nobel Prize for discovery of this system in bacteria. This is a pretty, simple systems. It's basically bacterial immunity, which protect bacteria from the uptake of the foreign DNA. And it's recognized self versus not self DNA. So how does it work? This system has 2 components. One component is called DNA methyltransferase, which can modify the DNA at certain DNA motifs. It can modify either adenosine to the 6-methyladenosine or cytidine to the N4 or N5 modification. Oh, okay. Okay, so, oops. Yeah. So we study in our lab the biology of this restriction modification system or technological part of this restriction modification system. I'm working for the company New England Biolabs, which is actually based on commercialization of this system for the research community to develop genetic tools for manipulation. And we produce these restriction enzymes and DNA methyltransferases as a product. Oops. So there are 4 types of restriction modification system known until this day. One, they are different by their genetic composition, their protein complexes, and their specificities. And there are known type 1, which consists of the 3 genes like restriction, endonuclease gene, metallase transferase gene, and specificity gene. And it's produce this type of the protein complexes, which can recognize usually interrupted sequence between some specific left and right arm of the motifs, and can modify M6 adenosine and M4 cytosine. The other, it's more commonly known for researchers, it's called type 2 system, which usually have only 2 genes, which are cognate genes, one for restriction gene and one modification gene. And they are produce separate peptides, which can recognize some specific motif like that and modify this type of modification. The third part, it's called type 2G, restriction modification system, which actually have all the functions on single peptide and produce single peptide which recognize non-symmetric, usually recognition sequence. And the type 3, it's similar to type 2, but it produces more complex protein makeup and also recognize non-palindromic sequence. While the type 2, usually it's a palindromic system. So in probably 2010, the company in California, which called Pacific Bioscience, start to develop a long-read DNA sequencing technology, which allowed to sequence long reads and compare, for example, with Illumina, which produce short reads. The advantage of long read technology, it's a mathematical problem where you try to assemble genome if you have the short reads. If you have some, and most of DNA have repeats, you cannot cover the repeats if you have only short reads. But if you have the long reads, it's much easier computationally to produce entire genome. So this technology, it's called smart sequencing, single molecular real time. It's working this way. So you produce the template, which is a fragment of DNA with bell-shaped adapters, and then you make a DNA polymerase complex on a bell, and then DNA polymerase begin to read like circles, and it produce like adapters, one strand, adapters, another strand, adapters, and so at the end you have a number of subreads accumulated. So all the sequencing error, it's actually like nivelated. So it has a very high level of accuracy, like about close to the 100%. And the all sequences is go on chips, which consist of like 150 for one instrument, up to modern system, it's 8 million deep holes in a chip, which cover and can bind the DNA polymerase at the bottom and entire DNA and DNA polymerase complex fixed at the bottom, and then fluorogenic nucleotides, one by one, incorporated, produce the light, which can be detected, like ATGC, and so you can get the sequence. The sequence can run for like from 10 to 30 hours. And because so much number of cells or wells, so you can collect entire sequence of entire organism in one run. And additional to just regular sequence, this technology also allowed you to use the so-called smart kinetics of incorporation of nucleotides to detect specific modification sequence, modified sequence. It's basically, this instrument can detect the time between the incorporation opposite to modified nucleotides. So if this A is modified, so then incorporation take much longer time than if this A is not modified. So, and this instrument can detect the basically epigenetic status of your DNA at the end. So this is just, I show the chemistry of the modified base which exists in bacterial species. It's a M5 methylcytosine, M4 methylcytosine, it has a little bit different chemistry, and MC adenosine modified. And the smart sequencing can see the signal of basically M4 cytosine and M6 adenosine, but it's difficult to detect of M5 cytosine because the signal is diffuse. But our company and like original study used some additional technology like bisulfite sequencing, which is a deaminate 5-methyl adenosine or cytosine, or like regular cytosine, and in the sequence this deadenylation, this process, deamination, produce instead of C, regular cytosines, produced thymidines, so you can read. And also our company developed a number of other techniques to detect this 5-methyl cytosine too. So how would we do this? So basically you have native host, which you want to sequence, and you purify the DNA, make a library, and for example, this host has like two metallases, like metallase R and metallase B, produce the library sequence, and then either on this, it's an old platform, and this is a new platform. And you have the sequence of this bacteria, as well the information about modified motif. But if you have several metallases in the genome, you cannot see which one is which. So in this case, we just purify the DNA, amplify the metallase gene, which we can detect bioinformatically, and clone this metallase gene, introduces a methylation deficient E. coli host, modify this host, purify the DNA from this host, and produce the sequencing result for each particular metallase. And we know which metallase, which pattern is modified. So this is the results of the detection of Prevotella intermediate from ATCC strain. We assembled this genome. This bacteria has a 2 chromosome and the number of different metallases, which shown here. And then, we clone one by one all these metallases and produce a table of which system recognize which motif. And in addition, we have these metallases, which can, in the future, help the researcher to overcome the barrier, because you can pre-modify it in E. coli with all these different metallases, or in vitro, the DNA of plasmid which you want to introduce to Prevotella. And in this case, it can overcome the bacteria. So it can trick the internal restriction modification system. So this is the result for the other clinical isolate of Prevotella 17F, which is also was sequenced and assembly. It also has a 2 chromosome, different sizes, and we detect all these different motifs which correspond different methyltransferase genes. And also, so this particular, for example, isolate can be engineered as a potentially to study, to make a genetic system for this Prevotella bacteria. We know the specificity, we can purify these metallases, so we can pre-modify the plasmids before introduction to this host. So it was not restricted anymore. So in addition to all this restriction modification system, when we analyze the assembled genomes of Prevotella, we find that both of these strains have this CRISPR-Cas system, which is also known bacterial immunity system. The 2 ladies got a Nobel Prize in 2020 for discovery of this system and engineers this system as a great tools for genome modification. I don't know, did you hear about that? But I mean, this system basically consists of the specific genes for the Cas proteins, which can use the tracer DNA or tracer RNA, and some RNA produced from this short sequences. These short sequences is actually the pieces of previous bacteriophages which infect this species of bacteria before, and it's introduced to the genome at the short palindromes, so they have like molecular memory for the bacteria about the previous infection. And if like new phage coming in, the bacteria can pick up any piece of RNA associated with these proteins and recognize and basically kill this bacteriophage, or like any other genetic elements which is a foreigner too. So it's another mechanism for bacterial immunity. So we find that both of these Prevotella has 2 CRISPR arrays, and on one chromosome, the other on another chromosome, and looks like complete Cas system to work that out. And the same thing with the other strain. And finally, I would like to make a conclusion of my talk. It's, in this work, we have utilized smart sequencing to define the complete genome, and for the first time, the metalome of 2 module Prevotella intermediate strains from ATCC and clinical isolate. And we identify and characterize multiple restriction modification system present in both, which support the hypothesis that this organism maintain a robust barrier to exogenous DNA uptake. And furthermore, we found considerable heterogeneity between strains, with only 2 out of 9 restriction modification systems shared between two strains. Additionally, in both strains, we note the presence of CRISPR system with consort with Cas protein, but no commonality in a protospacer sequence incorporated with the CRISPR array. And this work provided the critical resource required for development of genetic tools to overcome the genetic barriers of Prevotella intermedia, which will permit fundamental investigation into the organism physiology, metabolism, and pathogenesis in a human disease. And such tool will allow further mechanistic scrutiny of this pathogenic restriction modification and CRISPR-Cas system in future studies. And finally, I'd like to acknowledge people who work with me on this and other projects. There are people from New England Biolabs team which helped develop this 5-methyl cytosine detection system. My daughter, she usually helped me with editorial stuff. And our collaborator in a lab, these two persons. Also the people from Pacific Bioscience team, the guy who developed, he is the chief scientific officer of the company, our field representative, and our engineer, which have helped us smoothly run this smart sequencing platform. Also, our collaborator from the Harvard School of Dentistry and Forest Institute, and also New England Biolabs management, our scientific previous and current scientific directors, our department director, our CEO, previous and current, and the founder of the company. He died in 2020. And also there is a Rich Roberts lab. There are 1, 2, 3. It's bioinformatics scientists, and 1, 2, 3. Liquid wet chemistry scientists. Thank you for your attention. And if you have any questions, I will be glad to answer if I can. Yes, sure. Yeah.
Do you think that the same technique that utilizes this can be utilized with another bacteria from the orange complex and the mediated?
Yes, definitely. I mean, you can use this technique for any bacterial species. So we utilize, the people why came to us about that because we published a lot of genomes and a lot of metalomes on a different species for different projects. Yeah.
So it can be, if it can be replicated, do you think that by the modification of the sequences of the genome can have a direct effect on the receptor sites of the red complex species?
Yeah, I mean, if you can trick this restriction modification system, you can introduce the DNA to any species if you know what they are trying to destroy. So you know how to protect. So it's basically a question of self, non-self. If you can trick the bacteria to recognize the DNA as a self, so it can be accepted.